Okay, so welcome back to this next video in which we are discussing rheumatoid arthritis and anti-rheumatoid drugs. Okay, right, so we've just been discussing the adaptive immune response against a pathogen. Okay, what we now want to discuss is how the adaptive immune response is aberrant in rheumatoid arthritis and how this leads then to that initial infiltration of adaptive immune cells into the subintimal layer of the synovial membrane. Okay, right. So we're going to start off by discussing a major genetic risk factor for developing rheumatoid arthritis, okay, which is something called HLDR4 molecules, and I want to explain exactly what that is. Then we'll discuss the leading theory for how you develop this autoimmune response in rheumatoid arthritis, and we'll look at what citrullinated proteins are and potentially how they're involved, okay. Finally, then we'll talk about rheumatoid factor and potentially how that's not so involved, and maybe that's just something that happens in autoimmune diseases. Okay, right. So let's start off by discussing this major genetic risk factor then for rheumatoid arthritis. So the major genetic risk factor for developing rheumatoid arthritis is something called HLA-DR4, and I want to explain to you what this is. Now, this unfortunately is not ridiculously easy to understand. Okay, you need to have a decent understanding of the genetics of MHC Class 2 molecules to understand this. Okay, so we're going to talk then about the structure of an MHC Class 2 molecule in more detail than we have previously, and then we'll discuss the fact that you have 3 different loci on each of your copies of chromosome 6 which can make MHC Class 2 molecules. Okay, right, so let's start off by discussing the structure of the major histocompatibility complex Class 2 in more detail than previously. Okay, so the key thing that we need to understand is that MHC Class 2 molecules are made of 2 proteins, okay, 2 polypeptides stuck together. Okay, and let me draw these like so. Okay, so here is one of these polypeptides, like so, and it spans the membrane, and that's how it's attaching to the membrane. So these 2 parallel lines here, these are representing the cell membrane, so potentially the cell membrane of our antigen-presenting cells, so that dendritic cell, or the cell membrane of our B cell. Okay, and here's the N-terminus of this polypeptide, and here's the C-terminus of this polypeptide, and this is going to represent what's known as the alpha chain of MHC Class 2. Okay, and it has 2 important domains. One of them is over here, which I've shown as this curved region here. That's the alpha 1 domain, okay? And then the other important domain to know about is the alpha 2 domain here. Okay, right, so that's the structure of the alpha chain of MHC Class 2. Now let me show you the structure of the beta chain. So the beta chain, as far as my picture is concerned, it's just going to be the mirror image of this. Okay, so it's also going to have 2 domains, then a transmembrane domain, and then it's going to have its C-terminus intracellularly. Okay, so this is now the beta chain, the other polypeptide that makes up an MHC Class 2 molecule. And again, its extracellular domain is divided up into these 2 separate regions, one here in vivid purple, which is called the beta 1 domain, okay, and another here in turquoise, which is called the beta 2 domain. Okay, now, the peptide binding groove of your MHC Class 2 molecule is between the alpha 1 domain and the beta 1 domain here. So this is where loads of little peptide fragments can be bound and be presented to the T cell receptors of CD4 positive T cells. Okay, so this is the peptide binding groove. Okay, right, so that's all very simple. Now what we need to discuss is the genetics of MHC Class 2 molecules. Okay, and this is not simple. So basically there are 3 loci which can produce MHC class 2 molecules, and they're on chromosome 6. So the first thing to say is that you have 2 chromosome 6s. You have a maternal copy of chromosome 6, and you have a paternal copy of chromosome 6. Okay, we are now just going to have a look at one of these chromosomes, okay? And you're going to have the same 3 loci on your maternal and your paternal chromosome 6, although the alleles in these loci might not be the same. Okay, right. So, basically there are 3 regions on chromosome 6 which can all produce MHC class 2 molecules, and I'm going to now box these up like so. Okay, so these represent these 3 different regions. Okay, so these regions are known as the HLA-DP locus, the HLA-DQ locus, and the HLA-DR locus. So what does this mean? Well, firstly, what does HLA mean? HLA is just another way of saying MHC, okay? Strictly speaking, it stands for human leukocyte antigen, but this again just means MHC, major histocompatibility complex. Okay, so it's referring to the same thing as MHC, okay? Now, the MHC class 1 molecules, those are the HLA-A, B, C molecules. Okay, so if you ever hear someone talking about HLA-A, HLA-B, HLA-C, those are MHC class 1 molecules. The HLA-D molecules, those are MHC class 2 molecules. So when we talk about HLA-D, that means MHC class 2 basically. Now, it turns out that there are 3 different types of MHC class 2 molecules. There are the MHC class 2 P molecules, known as the HLA-DP molecules. There are the HLA-DQ molecules, and there are the HLA-DR molecules, okay, which are basically MHC class 2 proteins that are produced by these 3 different loci. So if you produce an MHC class 2 molecule from this locus, and note, I am not calling these pieces genes, okay? In fact, these areas contain loads of different genes, okay? So I'm calling them loci, okay? I suppose I should have more than one arrow since I've used the plural, okay? So these areas, DP, DQ, DR, they are loci, not genes. They contain loads of different genes, basically, okay? And in each one of these areas, or loci, you are going to have genes for alpha subunits or alpha chains of MHC class 2 molecules and beta chains. So in the HLA-DP locus, you will have genes for alpha chains and beta chains. Okay, and if you use those genes in this region to produce an MHC class 2 molecule, that MHC class 2 molecule will be an HLA-DP type MHC class 2 molecule. Similarly, if you use the alpha and beta chain genes from the HLA-DQ locus, then the MHC class 2 molecule that you end up with will be an HLA-DQ molecule. Okay, and if you use the genes from the HLA-DR locus for your alpha chains and your beta chains, then you will have an HLA-DR MHC class 2 molecule. Okay, and the other complicated thing is that each of these loci doesn't just contain one gene for an alpha chain and one gene for a beta chain. It's more complicated than that, basically. They contain multiple genes, more than 2 genes, and therefore they can code. Generally, usually it's only for one alpha chain, and then they've got multiple genes for beta chains. Okay? Right, so that's what we mean when we say HLA-DP, HLA-DQ, HLA-DR. These are 3 different types of MHC class 2 molecules. They all have this basic structure, okay, but the proteins will be different depending on whether you've used genes from one of these 3 loci, basically. Okay, so that's what is meant by HLA-DP, HLA-DQ, HLA-DR. The next complicated thing is that these regions are incredibly polymorphic. Okay, so what does that mean? That means that in the human population, there are a huge number of different alleles for these loci. So if you go around the human population and look at the HLA-DR locus, for instance, and I just pointed at DQ, I apologize. If you look at the HLA-DR locus, you will find loads of different alleles for this in the human population. That's why it's called polymorphic. Contrast that to other genes, basically, or other loci of the DNA where you don't find any alleles at all. There are some genes in the human genome that are identical in every single person on the planet, okay? I.e. there's no different alleles. But for these regions, they are hugely variable, okay? And in different people, you will have different alleles for HLA-DP, HLA-DQ, and HLA-DR, and therefore you will be producing different HLA-DP, HLA-DQ, HLA-DR molecules. So different people in the human population have very different MHC Class 2 molecules, basically, of the 3 different types. Okay, that is what is meant by polymorphic. Now, basically, there is a type of HLA-DR molecule that is called HLA-DR4, that if you have it, you are at very high risk or increased risk of getting rheumatoid arthritis. Okay, now, what does this mean? Is HLA-DR4 just a single allele of HLA-DR? Well, no, it's not that simple. Okay, firstly, you have to remember that HLA-DR contains multiple different genes. Okay, what HLA-DR4 actually refers to is it refers to a whole bunch of different HLA-DR molecules that loads of different people have, which are all quite similar, basically. They all have quite similar peptide binding grooves, okay? And they can bind similar peptides in those peptide binding grooves, okay? So MHC Class 2 molecules can present a huge number of different peptides, but obviously different MHC Class 2 molecules are going to prefer to bind different peptide fragments, basically. Okay, you can't imagine a system where it wouldn't be the case that they didn't have preferences for different peptide fragments. Basically, if we say that you have HLA-DR4 molecules, then what that means is that your HLA-DR molecules are of this certain type where the peptide binding groove likes to bind certain peptides, basically. Okay, so HLA-DR4 molecule does not refer to a absolutely specific type of MHC Class 2, i.e. you can't go onto the internet and find out the exact sequence of amino acids in the alpha chain and beta chain of HLA-DR4. No, that's not the way it is. There are loads of different alpha and beta chains that you can use to produce HLA-DR4 molecules. Okay, what is similar between all of these molecules that are classified as being HLA-DR4 molecules is that they all bind very similar peptide fragments. Okay, they have very similar peptide binding grooves. Okay, so basically, if you have HLA-DR4 molecules, that means that one of your HLA-DR loci, and remember you have 2 of them, one on each of the chromosome 6s that you have, is producing HLA-DR molecules that are of this type, basically, that have a very similar peptide binding groove and therefore will present certain types of peptide fragments, basically. Okay, so that's what it means. It does not refer to a specific allele. There are loads of alleles for HLA-DR loci that all are considered HLA-DR4, okay, because you produce HLA-DR molecules that are similar enough to be classed into this great umbrella term, basically. So understand that. Understand that if we say you are HLA-DR4 positive, what that means is you're producing HLA-DR molecules that have this certain type of peptide binding groove, basically. Okay, right. So, a certain type of MHC Class 2 then is a big risk factor for developing rheumatoid arthritis. And basically the reason we think this is because if you've got this type of MHC Class 2, you're more likely to present the peptide fragment corresponding to whatever antigen initially triggers the rheumatoid arthritis to your CD4 positive T cells. So basically it seems that this type of MHC Class 2 molecule, HLA-DR4, is capable of presenting a fragment of the antigen which is going to trigger the whole rheumatoid arthritis to the CD4 positive T cells, okay, and that's why it puts you at increased risk of developing this autoimmune disease. Okay, right, so now that we've got that discussion over and done with, let's talk now about the leading theory for how rheumatoid arthritis actually occurs. Okay, so now we're going to do the full picture at long last, or at least a theory, okay? It's not set in stone, this could be wrong, okay? So take it with a pinch of salt, basically, but this is the leading theory at present. The leading theory for how rheumatoid arthritis develops then is that it starts off with an infection, basically. You get infected with something, and we'll talk about a potential pathogen that might trigger it later on, okay? And basically what happens is this pathogen has an antigen that you launch an adaptive immune response against, and we will call this antigen of the pathogen, we'll call it the arthritogen. Okay, because it's what's going to trigger the rheumatoid arthritis. Okay, so the pathogen has this antigen which is going to have an adaptive immune response initiated against it, and that's perfectly fine. Okay, it's perfectly reasonable to launch an adaptive immune response against an antigen that is present on the surface of our pathogen. Okay, the problem then is that when you launch this adaptive immune response against this arthritogen here, okay, when you activate loads of CD4 positive T cells, and you also activate loads of B cells, producing loads of plasma cells which produce antibodies which combine to a specific epitope of this arthritogen, okay, the problem is, it turns out, we think, that adaptive immune response is capable of also attacking an autoantigen, okay? So basically we think that part of the arthritogen is too similar to an autoantigen, basically, okay? An autoantigen that seems to be particularly concentrated in the synovial joints, okay? So we think this is what basically happens. You get infected, you launch an adaptive immune response against a protein that this pathogen has, okay, and it turns out that adaptive immune response, unfortunately, can also attack a certain epitope of a self-antigen, an autoantigen, okay, that is present in the synovial joints. Okay, so the infection triggers the mounting of this adaptive immune response that then attacks the autoantigen, basically. Okay, so we think that the pathogen brings in this arthritogen that is just too similar, basically, to some of self-protein, basically, and then that adaptive immune response that you launch against the pathogen's antigen then ends up attacking the autoantigen. That's what our leading theory of what causes rheumatoid arthritis says. Okay, right, so we actually know a little bit more than this. We have a suspicion as to what this arthritogen basically is, okay, and we think that the arthritogen is some sort of citrullinated protein. Okay, so this is the next thing that I need to tell you about. I need to tell you about what a citrullinated protein is. Okay, so let's just discuss what a citrullinated protein is. Okay, so basically, arginine residues within proteins can be converted into citrulline residues after you have synthesized a polypeptide, okay? So let me show this. So I'll start off by showing you an arginine residue, and then I'll show you how it can be converted into a citrulline residue, and then we'll discuss what a citrullinated protein is and what it has to do with rheumatoid arthritis. Okay, so let me show you firstly the arginine residue then. So I'll start by drawing the core amino acid structure here. So here's the amino group, here's the alpha carbon with a hydrogen coming off it, here's the carboxylic acid group, and then the R group of the arginine, and I should just say that I'm drawing it as though it's an amino acid residue rather than the free amino acids. So I'm drawing it as though it's within a polypeptide. Okay, and the R group of an arginine residue consists of 3 methylene groups, and because I don't want to have to draw out 3 methylene groups, what I'm instead going to draw is 1 methylene group. Now I'm going to put brackets around it and then subscript it 3, and what this means is repeat that methylene group 3 times, okay? Then after the 3rd methylene group, what you'll have is an amino group, okay, which will be attached to a carbon, which has an amino group coming off it, like so, and then is double bound to a nitrogen, which then has a hydrogen coming off it. Okay, so this is the structure of the normal amino acid arginine. Okay, this is one of the amino acids that will have tRNAs corresponding to it, okay, and which can be put into a polypeptide as it's being synthesized. Okay, so if we draw our polypeptide here, it might have lots of arginine residues in, and the single letter amino acid code for arginine is an R. Okay, right. Now, what can happen to proteins after they have been synthesized is you can then modify arginine residues to a different type of amino acid that you can't just put in straight away because there are no tRNAs which put it in, basically. Okay, there are no codons which correspond to this amino acid. And this other amino acid is citrulline. Okay, however, it's an important component of some proteins. Okay, so let me show you the amino acid citrulline, rather. Okay, so once again, I'll draw it as a amino acid residue. So here is the core amino acid structure. So the amino group, the alpha carbon, the carboxylic acid group. And then the R group of citrulline consists of, again, the free methylene group, so it's very similar to arginine. Then you have the amino group there, then the carbon, and the amino group up here is exactly the same, but now that double bond to the nitrogen has been replaced with a double bond to an oxygen. Okay, so as I say, this is an amino acid that you can't put in straight away when you're synthesizing a protein, but you can convert arginines in your protein to citrulline, basically, after the protein has been synthesized. Okay, and this conversion of arginine to citrulline is carried out by enzymes called PADs. Okay, which stands for peptidyl arginine deaminase enzymes. Okay, so this stands for peptidyl, that's the P here, okay, and then it's peptidyl arginine deaminase. Okay, right. So this is a normal biochemical process. Some proteins in your body have arginine residues converted to citrulline residues, and proteins which contain citrulline residues are then called citrullineated proteins. Okay, now, in rheumatoid arthritis, we think that what happens is you lose immunological tolerance to citrullineated proteins, basically. We think that the autoantigen in the joints, whatever it is a citrullineated protein, okay? And we think that this arthritogen, which triggers the initial adaptive immune response that is brought in by this pathogen, is a citrullineated protein. So, to come back to this, what we think happens is some pathogen comes in with an antigen which has a citrulline residue within it. Okay, you then launch an adaptive immune response against the little fragment of the antigen that contains that citrulline. Okay, you produce T cells and B cells which are directed against this little epitope with the citrulline in, and unfortunately that then seems to be able to attack citrullinated proteins that are present in the synovial joint, but we still do not know what those citrullinated proteins in the synovial joint are. Okay, so basically we can find autoantibodies, well, not autoantibodies, but antibodies that bind to citrullinated proteins in 80% of people with rheumatoid arthritis. And this is very, very specific for rheumatoid arthritis. If you have antibodies against citrullinated proteins, it means you have rheumatoid arthritis, basically. That we cannot find these same antibodies in any other disease, which is not the same for the antibodies of rheumatoid factor, which I'll talk about later on. Okay, so basically we have a test which tests for what are known as anti-cyclic citrullinated protein antibodies, and I'll explain exactly what that means in a moment. Okay, so when we are suspecting that someone has rheumatoid arthritis and we want to confirm the diagnosis of rheumatoid arthritis, one of the tests you can do is test whether they have anti-cyclic citrullinated protein antibodies within their blood. So what does the cyclic mean? That's what we don't understand at the moment. The citrullinated proteins, the CP here, we understand, but what does cyclic citrullinated proteins mean? Okay, well this is basically just a technicality. Okay, so if we want to test someone and find out whether they have antibodies for citrullinated proteins in their blood, we need to test, we need to take a sample of their blood and test for antibodies that can bind to citrullinated proteins. Okay, so what do we then use as the citrullinated protein and to see if they bind to it or not? Okay, well, what you end up using is a little sort of cyclic polypeptide, basically, so a polypeptide that's wrapped around into a circle which contains a citrulline residue, basically. Okay, and this then is called cyclic citrullinated protein. Okay, so really, when we test to see if someone has antibodies against citrullinated proteins in their blood, what we are really testing for is if they've got antibodies which can bind to the cyclic citrullinated protein. Okay, and that's why, strictly speaking, we should say you are anti-cyclic citrullinated protein antibody positive, rather than just anti-citrullinated protein antibody positive. Okay, because it might be the case that you have antibodies that are directed against citrullinated proteins which for some reason don't bind to our cyclic citrullinated protein here. However, 80% of people with rheumatoid arthritis do have anti-cyclic citrullinated protein antibodies in their blood. They have antibodies that are capable of binding to our cyclic citrullinated protein here. And you do not find these in the normal population. In fact, you do not find this in anyone who does not have rheumatoid arthritis. So if we get a positive result for anti-cyclic citrullinated protein antibodies, we can be pretty confident that that person has rheumatoid arthritis, basically. Okay, right. So, going back to the pathology of rheumatoid arthritis then, what example of an infection have we found that can actually bring in citrullinated proteins then? Okay, because we have actually found now an example of a pathogen which has antigens which are citrullinated proteins and therefore could be an example of this arthritogen. Okay, right, so let me just talk about this example of a pathogen which has citrullinated proteins. Okay, right, so the example of a pathogen with citrullinated proteins is what's known as Porphyromonas gingivalis. Okay, so Porphyromonas, and then it's gingivalis. Okay, now this is a pathogen which is a gram-negative rod. Okay, so I'll draw a little picture of it here. It's a gram-negative rod, like so, and basically it causes very bad dental infections. Okay, not just gum infections, it causes what's called periodontitis. Okay, or rather periodontitis. Okay, so periodontitis. Okay, and what this means is an infection of the gums so bad that it is now actually spreading into the bone that attaches your teeth in, basically. Okay, so it's spreading into the bone underneath, so it's a very severe dental infection. Okay, now basically the idea is that you start off with an infection of Porphyromonas gingivalis causing periodontitis, okay, and this bacterium produces citrullinated proteins. It has been now found that it does have citrullinated proteins. The idea then is that you launch adaptive immune responses against these citrullinated proteins of this pathogen, okay, and then it turns out that these T cells and these B cells that you have activated as part of that adaptive immune response can then attack some autoantigen, can attack citrullinated proteins present within the synovial joint. Okay, and that's why then these CD4 positive T cells, B cells and plasma cells are infiltrating into the synovial joint at the beginning of rheumatoid arthritis. Okay, right, so that's an example of a potential pathogen that might be the initial driver for the development of rheumatoid arthritis. Okay, right, so I just want to give you one last piece of terminology with regards to this, and then I just want to mention rheumatoid factor. Okay, right, so another piece of terminology that people will often use is ACPA. Okay, and what this stands for is anti-citrullinated protein antibodies. Okay, so at the moment we think that the cause of rheumatoid arthritis is that you have produced these anti-citrullonated protein antibodies, okay, which are capable now of binding to some autoantigen within the synovial joints, basically. But they were initially produced to fight some pathogen, basically, that also had a citrullinated protein. So you think you start off with an infection that has citrullinated proteins as one of its antigens, okay? You then launch an adaptive immune response against those antigens, and that then causes you to lose tolerance for self-antigens, which are citrullinated proteins. You therefore end up with anti-citrullinated protein antibodies in the blood. Okay, but it's important to understand that there is a difference between anti-citrullinated protein antibodies in the blood and anti-cyclic citrullinated protein antibodies in the blood. Remember, the cyclic citrullinated protein antibodies, those are antibodies which are capable of binding to that cyclic citrullinated protein, okay? And we know that 80% of people with rheumatoid arthritis have anti-cyclic citrullinated protein antibodies in their blood, okay? But that doesn't mean that the other 20% necessarily don't have anti-citrullinated protein antibodies, okay? It means that they don't have antibodies which can bind to the cyclic citrullinated protein. It doesn't mean that they don't have antibodies that can't bind to some other citrullinated protein, so be aware of that, okay? However, I should now say that this is still very controversial, okay? This is hardly yet the set in stone truth, okay? So be aware of that as well. Right, okay, so the final thing I want to say is about rheumatoid factor, okay, which is one of the other tests we do to diagnose rheumatoid arthritis. Okay, so we test for rheumatoid factor within the blood. Now, what is rheumatoid factor? Well, rheumatoid factor is basically antibodies that are usually of the IgM type. Okay, so you have immunoglobulin M against the FC region, the constant region of IgG antibodies. Okay, so understand this. Basically, this is producing an IgM antibody, okay, which I'll draw here. So this is our IgM antibody, which is capable of binding to that constant region of IgG antibodies, like so. So this is our IgG antibody. Okay, so it's an antibody against antibodies, basically, against the constant region of IgG antibodies. Now, it's usually IgM molecules that you get as rheumatoid factor, but you can actually also get IgA type antibodies and IgG type antibodies produced that are capable of binding to the constant region of IgG antibodies. Okay, and these antibodies are known as rheumatoid factor, or RF, in the blood. Okay, and they are also something that you very commonly find in rheumatoid arthritis. You very commonly find the presence of rheumatoid factor in the blood. Okay, however, unlike anti-cyclic citrullinated protein antibodies, where they're not found in any other diseases other than rheumatoid arthritis, rheumatoid factor is found in loads of other autoimmune diseases. Okay, so if you have rheumatoid factor in your blood, it does not necessarily mean that you have rheumatoid arthritis. Okay, it most likely means that you have an autoimmune disease. Okay, so potentially rheumatoid factor isn't actually that important in the pathogenesis of rheumatoid arthritis. Potentially it's just a feature of autoimmune diseases. Potentially it's the body's response to antibody molecules that are causing disease. So in autoimmune diseases, it's the antibody molecules that cause the disease. So maybe now you're getting the immune system attacking the antibody molecules because they're causing a disease. Okay, who knows? That's still not well understood, basically. But rheumatoid factor is starting to sort of be shoved out of the way, basically, in favour of anti-citrullinated protein antibodies being the main thing which is involved in this autoimmune response. Okay, but it's important to understand that rheumatoid factor is one of the clinical tests that is done to diagnose rheumatoid arthritis, along with anti-cyclic citrullinated protein antibodies. Okay, right, and the one last thing I want to do is link this back to that HLDR4, okay, and put this all together as a big story then. Okay, so I said that HLDR4 molecules were a major risk factor for developing rheumatoid arthritis. Okay, so what seems to happen then is you start off with an infection. Okay, this has this special type of antigen which is called the arthritogen, which is capable of initiating an adaptive immune response which will then attack some autoantigen in the synovial joints. Okay, we think that this arthritogen now is some citrullinated protein of the pathogens. Okay, and basically that is now going to trigger an adaptive immune response. Now, where does the HLDR4 come in here? Well, it seems likely then that HLDR4 molecules are the molecules which must present the peptide fragment which contains the citrulline residue of this arthritogen to the CD4 positive T cells, i.e., if you don't have HLDR4, then it's much less likely that you're actually going to present that fragment of the arthritogen which contains the citrulline residue to the immune system and launch an adaptive immune response against that specific epitope of this arthritogen. Okay, so that is, we think, why HLDR4 is important here, that it's the molecule which is responsible for presenting the specific epitope of the arthritogen containing the citrullinated residue to the CD4 positive naive T cells. Okay, then you get the adaptive immune response initiated. You produce CD4 positive T cells that are active and also B cells that are active, which will produce an adaptive immune response against that specific epitope of the arthritogen. And unfortunately, it then turns out that they are capable of attacking a similar epitope in an autoantigen. Okay, and you break with immunological tolerance to citrullinated proteins, basically. And then that's why these cells end up infiltrating into the subintima of the synovial membrane in the synovial joints. Okay, so that now concludes our discussion of the pathogenesis of rheumatoid arthritis. In the next video, what we'll turn our attention onto is the anti-rheumatoid drugs.